Okay, so it's time for the next speaker. I would like to introduce Ahmed Ali. He's an assistant professor and he leads the single-cell research in a group of Thomas Hankemeyer. And he does this at LACDR, which stands for Leiden Academic Center for Drug Research. So I won't take any of your time. So
hi everyone, and thank you for the introduction, Tim. I have my timer. I know we're running a bit late. And I have 27 slides and 6 videos, but we'll do it. So, thanks again for the introduction, and I'm here to talk to you about single-cell metabolomics, and specifically untargeted metabolomics. I'll start by a very general overview on what I think are the main applications for single-cell metabolomics. And I think usually our experiments falls into one of 3 categories. You're either hunting for rare cell populations that otherwise you lose their signal if you do bulk analysis, or you're zooming in a little bit and you're interested for something inside the cell. Say, for example, you're looking for intracellular distribution of metabolites or drugs. And finally, the most annoying of them all, if you're studying the individual variations between the subpopulation, between certain subpopulations on the single cell level. Now, throughout this presentation, I'll take you through my journey and provide an example of each scenario, basically. And this is my first festival to attend, so I thought I would keep things light by also telling you about my very toxic, unhealthy relationship with single-cell metabolomics. So this theme will come back a lot during the talk. Now, a classic problem for rare cell populations is circulating tumor cells. Now, before I show you what we do with circulating tumor cells, I'll show you the method that we developed to target single cells. We would like to call it live single-cell mass spectrometry. Some call it capillary microsampling. The naming doesn't really matter. It is the Wild West, just like what Timothy said. How it works is relatively simple. So you have a capillary holder. You sample part of this very unhappy cell, or the whole cell if you want to. And then after this part of the cell is trapped at the very edge of the capillary. So you disconnect the capillary from the capillary holder, add an ionization solvent or an extraction solvent from the other end of the capillary. And because the capillary is half-coated with platinum, it can also act as a nanospray emitter for the mass spec. So I already can imagine the horror of the sample prep people and chromatography people here, because we do nothing. We just put the solvent and shoot everything into the mass spec and pray. It's a good method, I think, for what it does, but of course there is room for improvement, and that's why the love-hate relationship that I mentioned before. Now, going back to my example of CTCs, I'm not sure if you know CTCs or not, but basically they're very rare cells. They're associated with tumors. They are in the peripheral bloodstream of cancer patients. Now, the problem with CTCs is that they are very rare. So usually in 1 milliliter of peripheral blood of cancer patient, you have about 1 to 10 CTCs, and you literally have 1 million of other blood cells. So it's 1 in a million problem. With this in mind, we targeted neuroblastoma. So neuroblastoma is the most common cancer in children or babies back in Japan. The problem with neuroblastoma is that its symptoms are very diffuse. So fatigue, fever, etc. When my daughter comes back from daycare with a fever, I don't think of cancer, right? So it's very difficult to diagnose early. We collaborated a lot with Hiroshima University Hospital. We had the peripheral blood sample 5 milliliters of a 10-year-old neuroblastoma patient. And then we applied our technology. So we did FACS analysis in the beginning to sort the mononuclear cells. We did a lot of enrichment. And finally, we were able to isolate CTCs and macrophages in one dish. We labeled them both, and then we went in and sampled each individually. And we were very happy because we could find the same biomarkers that the physicians look for in the clinic. So physicians usually look for neurotransmitter biomarkers and metabolites in the urine of patients, and we could find these biomarkers in the single circulating tumor cell. So we were very happy about this, but then we thought about the next step. We can tell if a biomarker exists or not, that is already known, in a single CTC of a cancer patient. Do different CTCs from different cancer types have, well, of course they have a different metabolome, but is our method sensitive enough to detect these differences? And that's what we did next. So we targeted colorectal cancer and gastric cancer patients. This was in collaboration between 3 institutes, so Riken back in Japan, Singapore, and also the National Cancer Institute. We collected samples from both cancer types, and we tried a different isolation method that we think it's a bit gentler on the cells. It uses centrifugation, but I mean, we really don't know. And we could successfully, at least, see some differences, right? Now, I have to be very careful with my claims here, right? Because we only use MZ. Of course, we look at the isotopic pattern, etc. But at least it showed promise, right? We can see some features that are there with a very high significance, even after p-value correction, etc. Now, the problem that we had, and I think it was just mentioned, that we need more cells, right? We always need more cells. So we started a third collaboration, and we've been collecting samples for over 3 years now. We have 300 CTCs obtained from several cancer patients. And also, interestingly, the cells that, well, the tumor cells that the CTCs originated from. I can't show you anything yet, sadly, but hopefully next year I'll show you the results of this study. Now, switching gears a little bit, if we're talking about intracellular studies, one might ask, why bother, right? I mean, it's already difficult to do single-cell analysis by itself, and now you're asking me to measure things inside the cell, which is, of course, very hard. Now, the reason why it's important, if we're switching gears a little bit, is mainly due, I think, to drug discovery efforts. So the days where, if I'm a lazy scientist or whatever, and then I went on holiday and then I found some interesting mold and I discovered a drug and I get rich and famous are long behind us, sadly. Now, to develop a drug, it costs billions of euros. Now, the recent studies showcase that the overall success rate of a candidate molecule to reach all the way to FDA approval is about 10%. Understandably, drug companies, which are, of course, companies, so they're also driven by profit, are not very happy about that, right? So they published a seminal paper, I think maybe 3-4 years ago, where they identified 4 pillars. Why do they think that most drugs fail phase 2 clinical trials? And if you zoom into the first pillar, you're going to see that we need information on the subcellular level about where the drug is located, and preferably also how it's affected by the cell. So we need quantitative data. Now, this is where I've been my first slap with reality, because I was like, yes, let's use our method, it's great, we can identify metabolites from CTCs, et cetera, but then, of course, our method is not quantitative at all. It's purely qualitative, right? So it doesn't compute. We need to do something about it. So we collaborated with a Swiss company here in Europe. It's called Nanolive. They have a very interesting microscope. This microscope uses label-free technology to basically digitally stain the cell based on the refractive indices of different organelles or compartments in the cell. And what we did, let me see if the pointer works. Yes. What we did is we imaged the cell in 3D before we sample it, and then immediately we went in with our capillary and we sucked this part of the cytoplasm out, and this is how you get the shrunk part of the cell, right? So we took another 3D image after. Now, because all of this is digitally stained, the cytoplasm is stained, we can just subtract the images from each other, and that gives us quantitative data about the cell volume. Now, once you have the volume of the sample that you're measuring, well, not easy, but relatively speaking, it's easy to then use mass spec and do calibration lines, et cetera, and you quantitate. We did this twice with them. We also visited them back in Switzerland, and we drilled holes into their precious microscope to put our sampling setup on it. And we succeeded in showing for the first time using our method that there is indeed metabolic heterogeneity and different metabolic levels at different parts of the cell. That also made sense, of course, from the literature. I think Cell or Nature featured us, and at this point in my career, I was like, I'm going to be rich and famous. And I was very naive. It doesn't work this way in science, but it was very nice to be acknowledged, at least. Now, jumping again to the third category of experiments that you can do, which in my opinion are very, very difficult, is if you're investigating the individual variations between cells on the single cell level. Now, this is not just something that we just see under a microscope, right? Cellular heterogeneity, I think, has real effects. So a simple experiment that I ask my students to do is to just work with a relatively stable cell line, like, for example, here, MCF7, so it's breast cancer cells that are ER positive, stain them with an anti-cancer drug, tamoxifen, and then spike them with tamoxifen and then stain for apoptosis. And as you can see, not all the cells are affected equally by the drug. Many, many reasons why is this the case, of course, but I think that one of the possible things that we should investigate is if there is drug uptake heterogeneity between these individual cells. Do the cells metabolize the drug in the same rate? And are the cells affected by the drug equally or not? Now, I know this is, again, where I got hit, again, by reality, and I realized that mass spec, at least in my opinion, is incapable of doing this on the single-cell level reliably. Maybe I'm wrong, but this is just how I feel about it. Why is this the case? Because of two, or at least our method, of course, because of two intrinsic values. First of all, we're working with very small cells. So I work with mammalian cells in volume that, what, 10, 15 picoliters or something like that. And once I spray something into the mass spec using my method, it's gone. I can't really get it back, right? So our acquisition time is very small. I can't simultaneously try to quantify a drug, try to quantify its metabolite, and then measure the overall effect on the metabolome in one run in a single cell. It doesn't work. So what we did is we cheated a little bit, and I hope the mass spec people here don't throw shoes at me or something, because we used another method. So what we did is we had a cell, and then we look at the cell under the microscope, and then we shoot it with a Raman laser. Raman is another spectroscopy technique. I don't know if you're familiar with it or not. It's relatively non-invasive. I know we say shoot it with a laser and everything, but at least the cell is alive for a bit after we measure it with Raman. Raman is very good at getting a fingerprint, basically, of the cellular state. It's not selective at all, but this is all what we need to see if the cell is affected by the drug or not. And then, using our method for the same cell, we can sample it and then get more quantitative data for the drug uptake and drug metabolism. We designed a small experiment about this. It was published recently in Analytical Chemistry, so we're very happy about this, where we designed it around tamoxifen, because we want to measure three things: uptake, metabolism, and effect. Tamoxifen is metabolized by liver cells and into its more active form, which is 4-hydroxytamoxifen. And while it's metabolized, it's also toxic to the cells. So if we can succeed in measuring these cells, we have the trifecta of things that we're looking for, and we can try to get some accurate data about cell heterogeneity regarding these three processes. If we look to the Raman data, and I see I have 5 minutes, so I'll try to be more quick. If we look to the Raman data, we could build a after I saw the work by Timothy, very predictive, very naive model on if the cells are affected by the drug or not. We're very happy with it, of course, but I think we need more. We could isolate some, well, it's very difficult to do it with Raman, but we could isolate some biomarkers that also correlate well with the literature about what the Raman does to the cell. It initiates the apoptosis process, of course. Now, from the mass spec side, which is interesting for me because I'm a mass spec guy, is that we could reliably understand the abundance of tamoxifen across, well, many cells or dozens of cells. And what we could see, which was very interesting for me, that if you look to the left, you see that the relative standard deviation of tamoxifen compared to our control is almost 150% or so. Now, I'm a very paranoid person, and I thought, okay, maybe our method is bad. You know, it's just because of our method. In the same scan, we also measure 4-hydroxytamoxifen, which is its metabolite. And then you look at the percent standard deviation of 4-hydroxytamoxifen, and it almost doubled that. So what that makes me think, this is my hypothesis now, which is, of course, unproven yet, we're working on it, is that at each degree, you're going to find heterogeneity, in drug uptake and also in drug metabolism. We designed another experiment targeting this. We worked here with, at least from the mass spec, with not a lot of cells, about 36 or so, and I'll come back to this later. We designed another experiment, went back to Japan and measured about 400 cells. And frankly speaking, I've been scared to look at the data. So again, I looked to Timothy for this and his talk. It gave me a lot of courage, and I'll talk to him later about how do we correlate this data set. Now, in conclusions for Raman-MS at least, we were very happy because we could measure the uptake and metabolism of the drug on a single cell level. We're very proud of that. Of course, the data analysis, again, is a complete nightmare. And it gives me nightmares at night, actually. Now, we could predict the drug response. I could tell you with a high degree of confidence based on the Raman spectra and the models that we built that these cells are sensitive to tamoxifen or not, they're affected by tamoxifen or not. But we have very low throughput from the mass spec side. We analyzed about 240 cells by Raman, and we could only sample like a quarter of those and analyze them by mass spec, which is very bad, of course. We need to kind of have an even, of course, match between these two methods. So this is where I rolled up my sleeve and I was like, enough of this toxic relationship, and I'll dedicate my life to try to create the perfect single-cell untargeted metabolomics method. And we attacked it bit by bit. So I had a after I saw the Soleno robot, I feel very self-conscious about showing you this. But we had a very naive idea, is to just have a robotic arm replace what I do manually, which is picking up the cells. And I don't have a lot of time to show you this one, but I'll show you the next. But just quickly to go over it, you have the arm goes in, picks up a capillary. I'll switch to the next video quickly, and then brings the capillary into focus. And then after it samples the cells, it gives me back the capillary. And because I was having fun, I made the robot bow to thank me, because I was living in Japan. And they are very, very polite, and I like that. So that's nice. It's a good robot. So I'll show you the second video, which I think is cooler. We threw more money at the problem. We tried to also have high throughput. So we had two robots interacting with each other, trying to automate what we do. I sped up the video by about 20%. I apologize. The guy that's taking the video is not high or something on math or something. It's just sped up a bit, because I don't have a lot of time. You have the robotic arm again. There's a QR reader to keep track of the samples. And then because of the capillaries that we use, the length varies. So you have an X and Y laser that tries to tell you where exactly is the tip of the capillary. And slowly it goes into focus. Now it is very slow because I was doing a demo to high school children in Japan, and I broke 200,000 euros worth of equipment because I set it too fast and it hit the confocal microscope and broke it, and also the robotic arm. So I never do demos ever again. What you see here is also sped up a bit, is that we can control the robot via also kind of primitive GUI, to be honest, on the left. You move it up and down, left and right, etc. Once you have your cell, you push a button, and then it picks it up. Not fully automated yet, of course. This is just something that, our second attempt, right? After it picks up the cell, which will happen, maybe it will be too fast now. Let's see. Yep, too fast. After it picks up the cell, it goes to robot number 2. I see I already finished my time, but 1 more minute. It goes to robot number 2 via the very cool rack that we built. And then the whole existence purpose for this robot number 2 is just to pipette our ionization solvent. It's a very sad existence, but it's okay. It's science. We do it for science. So you have a very small capillary that goes inside our capillary and slowly pipettes the ionization solvent. It has to be glacially slow, because if we introduce any bubbles or air bubbles, that affects our nanospray performance later on, right? So this is why it's also very slow. I also sped this up. And then the rack goes back to the hero robot that does a lot of work, picks up the capillary. The capillary is still coated. And then you have very unsafe live wire with 1K volt applied to it. Please don't do this in your labs. We were doing things differently back then. And as the capillary touches the wire, the spray is started, as you can see here. Now, of course, this is not the final iteration of this, but it's just showing you what we did before. We're working on a lot of things as well. So from the data analysis side, we're attacking it with a wonderful group of colleagues that I've been working with lately. We're basically developing a whole new pipeline for how do we analyze our data. We called it SUMMER, which stands for Single-Cell Untargeted Metabolomics in R. We're very happy by the name. I did not choose it. Alida, our colleague, also did. We basically want to start from right where we get our raw data. So we use very high-resolution instruments. We also do a lot of tricks on how do we get even the data. So we really need to have a specialized solution. And then it will do the modeling, the visualization, everything, and also the alignment. Now, to integrate all the cool technology that you've seen here today, I'm also working with another equally wonderful group of people here. I think we have like 6 or 7 in total, so single cell, yay. That basically integrates everything that I talked about, and also adds to it the ability to do imaging at the same time, multimodal spectral analysis, and derivatization and cellular lysis in a controlled manner. Now this, we will not see this year, but hopefully in a year or 2, we'll have it also done. In the end, in the acknowledgement page, I want to acknowledge Arno. He's the Raman wizard that helped me with the Raman experiment. Yasmin, my colleague, and I'm very lucky to call her my wife, who's also helped a lot with the cell biology parts. The people that trusted me and I owe my scientific career to Dr. Sami Amara back in Egypt, Hiyama Sensei and Masujima Sensei, which is sadly not with us right now back in Japan. And of course, Thomas Hankmeyer, who invested a terrifying amount of money in me. So I have to deliver. Of course, the wonderful colleagues that help me wake up at night and be excited to go to the lab. Last and not least, my absolutely fierce daughter that kindly let me do this presentation and prepare it on the weekend, and her daycare for making an exception for her so I can actually present to you. I'll be happy to answer your questions.
So thank you for this fascinating talk. It was very nice and entertaining. I have a question about the intracellular analysis. So where you show your pipeline based on 3D holography, if I'm correct, and how that helps you guiding the harvesting of the different parts of the cells, let's say. So I wanted to ask you, how controllable is this harvesting? Like, can you harvest different organelles, for example? Can this replace ultracentrifugation?
So, some things are easier than others. If we're targeting different parts of the cytoplasm in different locations, that's easy, right? Because it's mostly aqueous or liquid. Different organs is a bit more difficult because we sample the cells while they're alive, like when we're just measuring them, right, by imaging. I think my colleague Yasmin only succeeded once in picking up the nucleus and leaving the cell intact. Now, with lipid droplets and things like that, it gets a bit easier as well because we can control the tip diameter of the capillary that we use. So we can go all the way from 1 micron to 10. So you can target things like that, but more difficult with nucleus, which is typically very solid, for example. But yeah, I'll say it's possible, but we're not there yet.
Okay, thank you.
Okay, thanks again, Ahmed.
Yeah, thanks.
And to conclude,